Welcome to Queer Science, Theology, Philosophy, and Spirituality. QSTPS, the Center of Science, Theology, Philosophy, and Spirituality, with an LGBTQ perspective. That's the song Natamani, meaning I Design Swahili, by Rebecca Dawn. Beautiful song. It is my song of the month. I love it so much. And being a musician, I'm gonna one day do it and blacken it. You know what we call by blackening. We make it sound like black gospel music and with a lot of oomph and power. Of course, it is a very nice song by an American Kenyan, and I enjoy it, and I said I'm going to do it my style, in respect of the beat of the song. It's a nice song talking about walking with the Lord. Anyway, back to our timeline, scientific timelines we're looking at. And we are studying today the history of genetics. Why are we doing timelines? We have said all science in the world, especially religion, are time-based systems. They all have times. They have time, especially majority of the written religions. Like Hinduism and Buddhism, most Eastern religions are cyclic. They are cyclical. They are, when you read their texts, they value cycles. And then when you come to the Abrahamic religions, Judaism and Christianity and then Islam, they are less cyclical. They are more linear in the sense that they have an end. They have a beginning and an end. Especially Judaism. Judaism will end when the Messiah comes. That's Judaism. And Christianity will end when Jesus Christ comes and then takes people to heaven. That's his Christianity. It has an end. There's no cycle. And then Islam also will end when the Mahdi comes and ushers in a time of glory and pomp. So that's why most religions, they have eschatology, they have timelines about life. And even the word timeline itself is created by people who value the linear aspect of time. And they are showing you that time travels in a line, not in a cycle. But in Eastern thought, especially Indian and Chinese, time is cyclical. That's why there's reincarnation. The Indians believe that you will be born, when you die, you come back to life in another life form, meaning you never die. You just come back as another life form, like that. That's Hinduism. And they're very cyclical. They believe the world always goes through cycles of destruction and construction. Destruction and construction. Of course, when Christianity has that aspect, there'll be a new heaven and a new earth. But it's not seen in a cyclical sense. It is seen in a linear sense. And then also science is also a time-based system. For information, science is a strong and the strongest belief system in the world because it is a belief based on facts. Most religions are belief systems not necessarily based on facts, based on faith. You just believe. You don't need a fact to believe. You just believe. That is religious belief systems. But science is a belief system that is based on fact. I believe the world is round because of the facts I have received, or the data I have received from the scientists who have traveled around the world, and the picture NASA has provided that the earth is round. You know, because someone can claim that picture is false. You see, that's the problem we're having these days. There are people who deny science. They even think, they claim scientists are lying. And how can a scientist lie? They are so, truth is science. If they lie, there is no science. They are not working. You see? So that's what happens. Now, so science is also a belief system that has a timeline. And in all the belief systems, science is the most linear, in the sense that they don't really value cycles. Even though cycles are actually scientific and happen in time, like depressions. You know, economic depression is a cyclic event. And you know, the reason why we have never had an economic depression is because we have data how to stop a depression. We know that when the world is going into depression, there will be a recession. So when a recession happens, it means that if you don't rectify the recession, you are going to have a depression. So the reason why the world has never had a depression is because it knows the symptoms of getting to a depression is a recession. So when the world goes into recession like it did in 2007 and 8, it rectifies. And that's what happened in the world. The World Bank and the governments of the world had to solve the issue that led to it. And then we found the problem. It was the subprime mortgages in America that led to the economic recession worldwide, apart from the America going to war in Iraq. There were very many factors. So that's the reason why there's no depression, and there won't be any depression. Yes, because we have now the data to stop. We know how to stop a recession. Therefore, you know how to stop a depression. It's like a disease. When you have HIV, there's a difference between HIV and AIDS, and a difference between HIV and AIDS. HIV is the beginning of succumbing to AIDS, but you can live your life with HIV and never get AIDS. Reason being, you have managed to control the virus with the ARVs. But if you're someone who is not controlling the virus with ARVs, there is a high likelihood that you can get AIDS, and so you succumb. So that's what I'm saying: recession to depression, HIV to AIDS. So that's what happens. Now, so that's the linear nature of time in the science system. And we're going to do this to many scientific fields. Now, we're on genetics today. So, what is genetics? Genetics is the study of genes. And what are genes? These are sets of chromosomes. These are parts of the chromosomes that make up human cells and nature, cells in nature. I was just seeing something today on the news, and they want to create a genome. You know, we already have the human genome. We've calculated all the genetic makeups of humankind. We now have the data. Now there's a new one to study the genetic data of all living organisms. That is beautiful, very beautiful. I can't wait for that, because now when we get that data, we are going to understand whom we are related to. Do you know humans are related to some organisms? Like the banana, 40% of the genetic material of the banana is the same with us. So what does that mean? It is wise to eat a banana, because it's just like you're eating yourself, 40% of yourself. Yes. And yet when you look at a banana, it doesn't look like you at all. It doesn't look like you. But it has the same genetic makeup. It's just like genetics about human beings. Do you know black people share genetic makeup with Arabs? There are Arabs who and Jews that share genetic makeup with Africans, black Africans. Yet the skin colors are different. So you wonder, how did that happen that you are sharing genetic makeup? You know, it is beautiful. So you can be a color, and that's why a blind person can be a white person, a black person, an Asian. You know, blindness is a genetic disorder, and it can be found in all races. So what does that mean? That genetic makeup can be found. Race is not necessarily genetic makeup. That's it. So basically that's what genetics is. And from our Wikipedia, they're telling us Hippocrates and Aristotle and Epicurus had started talking about genetics, our famous Greek guys. And there's a friar. Modern biology began with the work of the friar called the great Gregor Johann Mendel. He worked on pea plants and he published what is called Mendelian inheritance. So basically, Mendel was the person who helped us understand genetics. And then years went on and people created the genetic code and the gene expression. And in the 70s, we now had what you call genetic engineering. So are you seeing how science was evolving? Science is so progressive. And linear time is progressive. You know, something can, even diseases are linear, you know, much as they can be cyclic. A disease progresses, you know, sometimes it becomes worse or sometimes it reduces in gravity. Like when you take medicine and the disease reduces in gravity. You don't take medicine, the disease increases in gravity. So that's the linear nature or the oscillative nature of some diseases. These are cyclic, like the common cold. It always comes when it is cold or when there is a change of weather. The seasons are changing. You get a cold. You see, that's cyclic. So there are some cyclic diseases and they are linear diseases. And the easiest diseases to deal with are linear, because when you keep on pumping the system with medicine, it decreases the gravity of the disease and you may end up not having the disease. Like what has happened to HIV. Now people are undetectable, meaning the virus is so low they can't even pass it on to somebody, even though they are positive, because they have taken enough ARVs to crush any replication of the virus. So that is what you call a linear treatment of a linear disease. So genetics in science was a progressive, linear, getting better and better. From Mendel, it went to genetic code, then gene expression, then genetic engineering. Now we have genetic projects, and the scientists are even sequencing entire genomes. Genome is a genetic system. Beautiful. So let's go before pre-Mendelian ideas on heredity. Okay, these are pre-Mendelian. We don't want to focus on this. We are just focusing on timelines. That's all we are doing. Now, in the Charaka Samhita of the 300th century, that's 300 AD, ancient Indian medical writers saw the characteristics of the child as determined by 4 factors. Those from the mother's reproductive material, those from the father's sperm, those from the diet of the pregnant mother, and those accompanying the soul which enters into the fetus. These 4 parts creating 16 factors, of which the karma of the parents and the soul determined which attributes predominated and thereby gave the child its characteristics. So you can see the Indians were close. Actually, we have already talked about Hippocrates and Aristotle and Epicurus. they said they proposed the contribution of both males and females in the life of a child. So these are the Greek philosophers. Then after the Greek philosophers, Charaka Samhita, Indian medical writers, said there are 4 factors that determine the genetic makeup of a child. And this is what they gave: the mother's reproductive material, and the father's sperm, and the diet of the mother, and the soul which accompanies or enters the fetus. Then 600 years later, in the 9th century CE, the Afro-Arab writer Al-Jahiz considered the effects of the environment on the likelihood of an animal to survive. And in 1000 CE, that's 1000 AD, Abu al-Qasim al-Zarawi, also known as Al-Bukassis in the West, was the first physician to describe clearly the hereditary nature of hemophilia in Al-Tasrif. And in 1140 CE AD, Judah Halevi described dominant and recessive genetic traits in the Kuzari. So you see, people were going deeper and deeper and deeper. They were understanding genetics. And all this was before Mendel came. Now, in the 18th century, you now have Carl Friedrich von Gartner and Charles Nudin, who are doing hybridation, and they are creating hybrids. And then now we have Mendel. Mendel is the greatest geneticist before the double helix was found. And in breeding experiments between 1856 and 1865, Gregor Mendel first traced inheritance patterns of certain traits in pea plants and showed that they obeyed simple statistical rules, with some traits being dominant and others being recessive. These patterns of Mendelian inheritance demonstrated that application of statistics to inheritance could be highly useful. They also contradicted 19th century theories of blending inheritance, as the traits remained discrete through multiple generations of hybridization. I like that there are some traits which were hidden because of hybridization. All right, so Mendel discovered that there were patterns, and he discovered that they were so mathematical. They were obeying mathematical rules. So what does this tell you? I believe in biology, everything is mathematical. Everything in your body is mathematical. So we have to continue counting. And now, post-Mendel. This is what happened in post-Mendel. Post-Mendel, meaning after Mendel, these guys came with their theories. Darwin is one of them, who came with the theory of natural selection and the theory of heredity, pangenesis, and others. And then in 1883, August Weismann conducted experiments involving breeding mice whose tails had been surgically removed. So there you have it. Now let's go through let's go through the timelines, and then we will be done with the video. So in 1856, 1863. So what does this tell you? The genetic science timeline starts in the 19th century. And here we find Mendel, and he established the principles of inheritance, namely principles of dominance and independent assortment and segregation. Then in 1866, he wrote about plant hybridization. Then in 1869, Friedrich Mischer discovered a weak acid in the nuclei of white blood cells today we call DNA. Congratulations to Friedrich Mischer. And he isolated the cell nuclei and separated them from bandages and then treated them with pepsin. From this, he recovered an acidic substance which he called nuclein. And you see, DNA is an acid. So Frederick found it but he just called it nuclein because he found it from the nucleic cells. But he didn't know what it was he had found, and he just called it nuclein. Then, in 1880 to 90, Walther Fleming and Edward Strasburger and Edward van Beneden elucidate chromosome distribution during cell division. So these guys found that chromosomes are distributed when the cells divide. Remember, fertilization involves cell division. And so these guys said chromosome distribution happens, and chromosome contains DNA. So that's when your parent's material is distributed to the person, to the zygote, because that's what cell division, fertilization is actually cell division. Now, in 1889, Richard Altman purified protein-free DNA. However, nucleic acid was not as pure as he had assumed. It was determined to later contain a large amount of protein. Now, another thing about protein you need to understand. What is the common word you know for protein? Protein is a body-building food. Protein is, we used to call it in children, baby science, we called proteins body building food. So proteins are body builders, and therefore DNA builds us up. Now, in 1889, Hugo de Vries postulates that inheritance of specific traits in organisms comes in particles such as pangenes. And in 1902, Archibald Garrod discovered inborn errors of metabolism. And an explanation for epistasis is an important manifestation of Garrod's research. When Garrod studied alkaptonuria, a disorder that makes urine quickly turn black due to the presence of Gentestate, he noticed that it was prevalent among populations whose parents were closely related. So you see, Garrod discovered inborn errors of metabolism. What is metabolism? It's a breakdown of chemicals. So some people make urine. Their urine, instead of it becoming yellow, it turned black because of the presence of Gentesate. And he found this only to be prevalent in parents that were closely related, like cousin inbreed, inbreed marriage and inbreed reproduction. So that's what happened, and he found out that genetics was to blame for this black urine that these people were having. And then Walter Sutton and Theodore Boveri hypothesizes that chromosomes which segregate in a Mendelian fashion are hereditary units. So these guys said that any chromosome that separates in the way Mendel had explained, those are hereditary units. They are passed down, and pairs that chromosomes occur in matched pairs of maternal chromosomes which separate during meiosis. He concluded that this could be the physical basis of the Mendelian law of heredity. So what he's trying to say is that the mother has her chromosomes that are hereditary, and the father has chromosomes that are hereditary, and they separate during meiosis. There is mitosis and meiosis, which is now the separation. So these guys were just confirming Mendel, and that's why it's called the chromosome theory. Now, in 1905, we have now entered the 20th century. So let's look at how many things happened in the 19th century. 1, 2, 3, 4, 5, 6, 7, 6. 6 major events happened in the 19th century. And then we're going to count how many events happened in the 20th century. then we'll know where work was done. And so, in 1905, William Bateson coined the term genetics in a letter to Adam Sedgwick at a meeting in 1906. Can you imagine? Many years ago, the genetics was born, the word genetics. And then in 1908, these two men, Hardy and Weinberg, they came out and proposed a model which describes the frequency of alleles in the gene pool of a population, which under certain conditions disturbing are introduced. So there are certain alleles, the frequencies of alleles, parts of the DNA, they are created or are introduced because of certain specific conditions and in every generation. So that's what this Weinberg and Hardy brought. Then in 1910, Hunt Morgan showed that genes reside on chromosomes while determining the nature of sex-linked traits. So Morgan now found out genes live in the chromosomes while sex-linked traits are determined. And how did he do this? He did this by studying the fruit fly, Drosophila melanogaster. See, the black melano is there. And the white-eyed based on Mendelian's principle of segregation and independent assortment. So I want you to understand that on Mendel's discovery and principles. So Mendel is the one who opened the door of genetics. He started, and then the father of genetics, you could say. And it's his principles that helped new geneticists come up and study what he found out. And this is the beauty of science and scientific people, is that they go into a house. Remember, genetics has been in the world ever since nature, life forms have existed, especially biological, that is beings that have water in them. They have genetic makeup. They have existed for thousands of years. But very few people sacrificed their time to study what this thing was. That's why we honor scientists through the Nobel Peace Prize and very many rewards. That's why countries that are great economically they value their scientists. They value their universities. America is a great nation because of the university military government complex. They were all connected. The military worked with the universities, and the military is a government wing. So research, the universities were doing research for the government and the military. And that's how America grew. Everything, even business. Can you imagine? People think, why should I go to school to study business? My friend, there is a lot of data you need. And data is only found when research is conducted. And so when a university conducts great research, it will produce great data for the government to implement policies that will produce the models that the scientists in the universities have found. If they are implemented, it will lead to economic development. And that's what happens when you connect with universities and scientists. Now, 1911, Alfred Sturtevant, one of Morgan's students, you see. So what is also happening is what you call academic generations. You don't need to have a child physically to pass down knowledge, to pass down something, to pass down your genes. You can pass down your academic genes. Mendel passed down his academic genes by writing what he studied. Then Morgan read it and understood it. Then Morgan taught Alfred, And then Alfred invented a procedure of linkage mapping, which is based on the frequency of recombination. So you see, the great-grandfather of Alfred is Mendel, academically speaking. And that's the same even in religion. You know, the father of Paul is Jesus. Paul even called himself adopted because he didn't walk with Jesus. And then Timothy calls Jesus his grandfather theologically because the teachings of Jesus were passed down to him through Paul, who was the son, adopted son of Jesus. So that is what you call theological generations. And now we are talking about scientific academic generations. You pass knowledge on to you don't have to have children to pass down genes. You can pass down various types of genes: academic genes, theological genes, economic genes, philosophical genes, like Karl Marx. The child of Karl Marx is Lenin. Lenin took Karl Marx's teachings. Then the famous Mao Zedong of China is a grandson of Karl Marx. And Savimbi of Africa. All of these socialist revolutionaries, they were children of Karl Marx. So that's what I'm trying to show you. With 1913, and he made the first genetic map of a chromosome. And in 1913, the gene maps showed containing linear arranged genes. So you see, there you are. And Ronald Fisher published correlation between relatives on the supposition of Mendelian inheritance. See, Mendel was still disturbing people in 1918. And then Lysen-Koism started. During Lysen-Koism, they stated that the hereditary factor are not only in the nucleus but also in the cytoplasm, though they called it living protoplasm. So the cytoplasm is a part of the cell. When you look in the cell, there's something called a cytoplasm, and it is also part of hereditary. It also determines hereditary traits, and that's what was found. And then in 1923, after World War I, Frederick Griffith studied bacterial transformation and observed that DNA carries genes responsible for pathogenicity. So, genes that create pathogens are in the DNA. Very important thing discovered by Frederick Griffith. Very important. This is where we get the idea that diseases are hereditary. Disorders are hereditary. Even now we are going to find out sexuality is also hereditary. Yes. If you find gay children in your family, somebody who was gay, in that chromosome, in that DNA, yes, it is scientific. It is scientific. And as I said in my other videos on my other channel, Rex Arthur, that we will do genetic. It's a fact. There are some races that have more homosexuals than others. It's a fact. But it doesn't mean homosexuality is exclusive. It is universal because it is a biological trait, meaning anybody who has life can be homosexual. Even that's why there are animals which are homosexual. It's because they are biological beings, so they are susceptible to homosexuality. And that's why lions, chimpanzees, giraffes, they also exhibit homosexual behavior. They are biological like humans. That's why we are animals. And that's what people need to understand. We are animated. And we are animated because of blood and fluids and gases working in us. Hallelujah. All right. And Frederick goes on and he discovers that hereditary material from dead bacteria can be incorporated into bacteria. In 1930s to 50s, Joachim Hammerling conducted experiments with acetbularia in which he distinguished the contributions of nucleus and cytoplasm, later to be discovered DNA and mRNA, that's ribonucleic acid, respectively, to cell morphogenesis and development. So Joachim now helped in studying to distinguish nuclear from the cytoplasm, and what was in the nucleus was called DNA, and what was in the cytoplasm was called mRNA. 1931, there was crossover is identified as the cause of recombination, and this was done by Barbara McClintock and Harriet Creighton. Look at those two women working. Maybe they were lesbians, we don't know. And then 1933, Jean Brachet, while studying virgin sea urchin eggs, suggested that DNA is found in the cell nucleus and that RNA is present exclusively in the cytoplasm. So RNA, ribonucleic acid, is exclusively found in the cytoplasm, and DNA is found in the cell nucleus. Very important discovery by Jean Brachet. Now, Thomas Morgan received a Nobel Prize for linkage mapping. Congratulations to Morgan, the son of Mendel, academic son, not real son. And his work elucidated the role played by the chromosome in heredity. So you see, Nobel Prize have been around for quite some time, and they study and they honor the work of scientists for showing us linkage mapping. And in 1941, Tatum and Beadle showed that genes code for proteins, and they, of course, codes, meaning, you can code them down, you can write them down. And Luria-Debruck experiment showed that genetic mutations conferring resistance to bacteriophage arise in the absence of selection rather than being a response to selection. Good. Now we come to the DNA era, and that's 1944, and they isolated the DNA as a genetic material. And Salvador Luria discovered the reactivation of irradiated phage, stimulating numerous further studies of DNA in the bacteriophage. And then Barbara McClintock discovers transposons in maize. And then Chargraf determined the pairing method of nitrogenous bases. So this is a lot of science here. We're just touching, and you can see Hershey Chase, again, 1952, proves the genetic information of phages and by implication all other organisms to be DNA. So you see, this is what they showed: DNA. These are the bacteriophages, and all of them have DNA. And beautiful that today, as I told you, that news item, we may check it if we finish the video quickly, that he's saying they wanted to find out all the genetic data of the life on Earth. So you can see infection, blending, and centrifugation. So what was the work of this experiment? That genetic information of phages and all other organisms is DNA, deonucleic acid. So organisms are naturally acidic. Go and pour an acid on any living organism, including a human. You will see. It will change. Reason, we are acids. So when you put an acid on another human being or another life form, it will change it. So, and the most important year in genetics, in my view, is 1952 and 1953, when an X-ray bifurcation image of DNA taken by Raymond Gosling in May 1952, who was a student supervised by Rosalind Franklin, and the DNA structure was resolved to be a double helix by Rosalind Franklin, James Watson, and Francis Crick. To me, 1952 was the most important time because now we knew how the DNA looked like. You know, at the beginning it was just fluid. People didn't know what it was. But now the structure was seen, and thanks to, very important, X-ray. Meaning, the time of Mendel, there was really no X-ray imagification. So they couldn't know what it looked like. So they were limited. And this is the thing you need to understand about science. That when you study things, you may not know everything in the time space you have. Reason being, you don't have the tools to decipher what something is. Actually, anything in science that you do not know right now, it's because you don't have the tools to decipher it or to study it well. But the moment you get the tools, believe you me, you will study these things. This is the reason why these things were discovered in 1952 and not in the year 1498, you know. Because in 1498, there was no X-ray, there was no, microscope. People were just hanging there. They were just trying to survive. But when the tools come, the opportunities, when the environment is conducive to conduct scientific research and to know something, you will know that thing. So that's why there are things we don't understand. We don't know everything for information. We still don't know everything. And one of the reasons is we don't have the tools that we could use to know that thing that we do not know. So Todd, then after the DNA was drawn very well, you had Alexander Todd determine the chemical makeup of nitrogenous bases. And he won the Nobel Prize, determined the number of chromosomes in humans to be of 46. You see what happened after the structure of DNA was found? Immense discoveries now. People now started knowing what was part of DNA, and they found the nitrogen, and they found the adenosine and triphosphate, and then after that, they counted the number of chromosomes in humans to be 46. And then counted all of these. Then Arthur comes in with Severo Ochoa and then synthesized DNA in a test tube, after which discovering the means by which DNA is duplicated. They got to know DNA is duplicated, and they won a Nobel Prize. Just look, Nobel Prizes were just flooding in the 50s, and it was based on genetics. So it tells you in the 50s, genetic science exploded. And then you have Hawley, Nirenberg, and Corana. They proposed the nucleotide sequence of tRNA. And guess what? And this is showing the experiments of Stahl, that DNA is semi-conserved replicate. So understand that DNA is repeated. You see, it's cyclic. Jake covered a group of genes whose expression is coded by an operator. And then Bren and Crick found shift mutations, taking shifts. And then you have Messelon and Jacob and Brenner. They identified the function of the messenger, RNA. All of these things happened. Look at the cracking, the genetic code by Corana and Neren, Brenner and Crick. Combined, they were excited. They were so busy, and they were cracking the code. And all of this happened. 64. RNA viruses, that the direction of DNA to RNA's transcription can be reversed. Are you seeing? Most viruses are genetic. Good. And then, like lysenkosim ends. And then they crack the genetic code by using RNA. Again. And heteropolymers and homopolymers. There you are. People who think there is no homosexuality is there. There is homosexuality and there's heterosexuality. Because in science, there is the principle of homo and the principle of hetero. It's like twins. You have identical twins of the same sex and fraternal twins of different sexes. 1970, restriction enzymes discovered. And then Stanley and Herbert, they constructed a recombinant DNA. And then now we come into the genomics era. And that was, look at that, beautiful. Oh, look at that art, scientific art. The first gene that was sequenced in 1972, the gene of bacteriophage MS2 coat protein, and with chains in different colors. Are you seeing the chains? Beautiful science. It's unbelievable. And they sequenced a gene. And then Walter fires and determined the complete nucleotide bacteriophage MS2 RNA. And then you have 1976, yeast genes were expressed in E. coli for the first time. Can you see? Yeast is also genetic. DNA is sequenced for the first time. And remember yeast, for all you Bible people? It was genetic. They didn't want yeast genes to be on the bread. It's beautiful. 1977, remember Sanger? Oh, sorry, I thought it was a lady. Fred Sanger and Walter Gilbert and Alan Maxam, they sequenced DNA for the first time. Unbelievable. You know what is to sequence DNA is to study the entire genome, the whole structure of DNA in a human being. That's what DNA sequencing is all about. And that happened in 1977. And Berg, Sanger, they developed methods of mapping DNA, the structure of DNA. And then Cohen and Boyer received the first US patent for gene cloning, proving the successful outcome of a cloning plasmid and expressing a foreign gene in bacteria to produce protein foreign to a unicellular organism. So there you are, 1980, gene cloning happened. 1982, insulin. You see, we now entered genomics. People stopped studying what DNA looked like, and now they were creating fluids, and the first was the genetically engineered human insulin, originally biosynthesized using recombination DNA methods by Genentech, 1978. And in 1983, invented through the polymerase chain reaction. And Barbara for that long. 1983, she won the Nobel Prize in Physiology or Medicine for her discovery of mobile genetic. See? She moved. Wonderful. We have Alec Jeffrey's method, and he studied DNA variants and the evolution of gene families in order to understand these causing genes. Many diseases we have are genetic. So that's what you do. You solve, if you want to cure a disease, 80%, you will cure it genetically. Now, Jeremy Nathans, look at 1986. Genes for color vision and color blindness. And they were studying the complexity of the retina. You see, accidentally, you will be studying something else in science, and then you fall onto the fact that color vision and color blindness is genetic. Yoshizumi accidentally discovers and describes part of a DNA sequence which is called CRISPR. And Thomas Cech discovered RNA can catalyze chemical reactions, making one of the most important breakthroughs in molecular genetics because it elucidates the true function of poorly understood segments of DNA. So RNA works hand in hand with DNA. And then 1989, human genome encodes CFTR protein sequenced by Francis Collins. And then they found that cystic fibrosis is genetically caused. And 1992, American and British scientists unveiled a technique for testing embryos in vitro, amniocentesis, for genetic abnormalities like hemophilia. You see, hemophilia that was discussed by our Arab friends up here in hundreds of years, the hemophilia that was discussed by Abu Qasim al-Zarawi, you see, is, sorry, I've lost where I was. Let me go back. What do you call it? I was talking about the hemophilia that he discussed. Was now found to be genetic 600 years later. 600 years later, it was found to be genetic. This is where we were. I was trying to tell you. Abu al-Qasim al-Zarawi was the first physician to describe clearly the hereditary nature of hemophilia. So he could only describe that it is hereditary, but he didn't know what was happening. He couldn't explain it. But now come years later, 600 years later, over 600 years later, 1990, in 1992, is it 92? Yes, 92. The American and British scientists, they found the genetic abnormalities such as cystic fibrosis and hemophilia. Beautiful. After all those years, they now discovered and confirmed Zarawi's theory. 1993, Roberts awarded the Nobel Prize for discovering that genes are made up of introns and exons. Then 1995, Haemophilus influenza. There is the first genome of a free-living organism to be sequenced. And more yeast, 1996. And in 1996, Alexander Rich discovered the zDNA. It is a type of DNA which is transient state associated with transcription. 1997, a very important year in genetics for me. I remember it very well. Dolly the sheep was cloned by Wilmot. And this was the most important because a sheep was created. People attacking scientists that they are playing God, you're late. They already did it long. And they cloned Dolly. And in 1998, the first genome was sequenced for a multicellular eukaryote, and it was released now in the year 2000. So we have entered the 21st century. Drosophila melanogaster, full genome sequence completed. And remember, we saw it here in what year? 1906. Let's see. Is it 1906? Yes, 1910. Thomas Hunt Morgan showed that genes reside in the chromosomes when he was studying Drosophila melanogaster. And now look, in the year 2000, the full genome Drosophila melanogaster is completed. So what am I trying to say? That science is a long study. This is 1910, 90 years later. They did a full sequence of Drosophila melanogaster to tell you that science values long study. When you're a scientist, one of the things you have to have is patience. And in this fast-paced world, actually, right now, what AI is doing to science is hastening very many things, like sequences. Do you know these days you can now do very fast genetic genome sequences? Reason being, we have AI, we have all of these, we have supercomputers. So a supercomputer is able to calculate and able to quantify matter very fast. And we are headed there. We are having now supercomputers, and now you feed them with data, genetic data, and they're able to analyze it very fast in the quickest way. And the faster it is, we're going to get treatments and cures in a jiffy. You know, it's that fast. We have to use the word jiffy. So it's a wonderful world. 2001, first draft of sequences of human genome is simultaneously released by the Human Genome Project and Celera Genomics. And Francisco Mujica and Ruth Jansen in 2001 created CRISPR, the acronym, to describe the family of bacterial DNA sequences that can be used to specifically change genes within organisms. And 2003, there was a successful completion of the Human Genome Project, with 99% of the genome sequenced to a 99.99% accuracy. Look at that. And 2004, Merck introduced a vaccine for human papilloma virus which promised to protect women against HIV, HPV 16 and 18, which inactivates tumor suppression genes and together causes 70% of cervical cancers. So you see what was happening? Now that the genome was sequenced and everybody understood DNA and everything, people were cured. The vaccine came for HPV. Why? Because viruses are genetic. Never forget that. Even right now, HIV is said to have been cured because through the genetic editing. It is happening, and it's gonna increase. 2007. Look at that. Oh, as if I was reading this thing. 2007, Michael Warraby traced the evolutionary origins of HIV by analyzing its genetic mutations, which revealed, beautiful sentence, that HIV infections had occurred in the United States as early as the 1960s. So, Michael discovered the evolutionary origins of HIV. It is evolutionary. Why? It's a genetic disease that was mutating. By analyzing its genetic mutations, genes mutate, and a genetic disease also mutates. And that's what HIV is. And Michael Warraby found out. And look what happened. The same year he did that, Timothy Ray Brown becomes the first person cured from HIV/AIDS through a hematopoietic stem cell transplantation. You see, stem cell medication is now working, and this is the 21st century. And you see how fast people got the way to deal with it? It is because of the knowledge. When the knowledge is so much, you can solve problems very fast. If you know a car very well, you know the parts of a car, let me tell you, you can fix the car within 5 minutes because you know the problem. This is why I always tell people, please study how a car is made so that you can know how to fix it fast. You will not sleep there 10 years fixing a car. Because you will sleep for 10 years fixing a car because you don't know how it works. It's a fact. But if you know how a car is fixed, how a phone works, and how a computer works, believe you me, there is no problem that can beat you, because you will know where the problem is and what needs to be solved. And that's what has happened. That's why HIV can be treated. It's not because it's because you know the nature. It's genetic. 2008, Houston-based Introgen developed Advexin, the first gene therapy for cancer. Look at that. Now cancer is even being treated. And Lifromena syndrome, utilizing a form of adenovirus to carry a replacement gene coding for the p53 protein. So you see, people are busy now because they have realized even cancer gene therapy can cells have nuclei, which are DNA. So cancerous cells have genes, and to fight them, you use gene therapy. The chemotherapy is the use of radiation to destroy cancerous cells. But gene therapy goes to the genetic root. And to cure any disease, you always go to the genetic root, because the gene is the smallest unit of the cell. The cell is the smallest unit of the body, but the gene is the smallest unit of the cell. So if you want to cure a cell, go for the gene. And this is what happened here. And the first gene therapy for cancer. Transcription nucleases or talents are first used to cut specific sequences of DNA. And in 2016, 2 years ago, a genome is sequenced in outer space for the first time, with NASA astronaut Kate Rubins using a mini ion device aboard the International Space Station. Congratulations, Kate. You sequenced a genome outside the world. So you see, we're even having fun now. We can even sequence genomes in Mars if we want. And that's beautiful. So thank you for joining us. You see that we'll go through history of molecular biology one time and all of these others, but today we wanted genetics, and it's beautiful. So what have we learned from all of this? We've learned one thing, that science is progressive in time, science is linear, and what do we mean by progressive? More knowledge increases with time. You see, we start with the basics, then we go deeper and deeper, and when people discover more knowledge about the thing, we build on that knowledge and discover. That's why people are getting better and better and better in understanding something until the lifeline of that thing is done. Do you know there's some topics in science that have closed? Yes, they are closed. The closed chapter. Meaning, because they have a lifeline. A timeline is a lifeline. There are some topics in science that have already ended, like motion. Newton's laws of motion. It closed the chapter on motion, in the sense that there will be no other law you cannot undo the action. For every action there's a reaction, because it is a law. And the beauty of scientific laws, they cannot be amended. We can amend human laws. Human laws are amendable. They are social. They are so flexible. But scientific laws are laws based on nature. They cannot be amended. That's what you need to understand. So genetics science has gone that far, and it all started with one man called Mendel. And not only Mendel, even before Mendel, the ancient philosophers. You see, that's why we are also a philosophical center. Philosophers talk about everything and anything. Aristotle, Hippocrates, the doctor, they talk about everything, and they gave their views, and that's what has happened. Okay, guys, thank you so much for joining us on the genetic science timeline.